Hello,
Hello?
everyone. We're gonna start with our last session of talks for plants and microbes. Uh, I ask you not to make questions on the chat now, just when I let you know when it is questions time, so you can use the chat and unmute yourself if you prefer. And we're gonna start now with Andrea Vilamizar. So, Andrea, you can share your screen so we can see your slides. Thank you. Now I'm gonna mute myself.
Hi, is everybody seeing the screen?
Yes, we are.
Okay, cool. Then I'm gonna minimize this. Hi, well, thank you for coming to my talk. My name is Andrea. I am going to talk to you about the distribution of this pathogen that has been killing frogs. Oops, sorry, for the past um, 100 years. I also would like to acknowledge my co-authors first, Dr. Juan, Dr. Castro, and Dr. Forshner, because I could have done this without them. So amphibian populations have been declining for the past 100 years, and there are three major drivers for this declines. We have habitat fragmentation, pollution, environmental changes, and emergent infectious diseases. And one of these diseases is called Batrachochytrium dendrobatidis. And I know it's a mouthful, so from now on, I'm just going to call it BD. BD is actually a fungal disease, and it invades the amphibian skin. And what it causes is it prevents amphibians from doing an electrolyte exchange with the environment. And in the end, it just causes death. The problem with this disease is that it's actually considered to be the worst disease affecting wildlife right now because it affects over 600 different species of amphibians all over the world. And there are two possible hypotheses of how this disease came to be in the United States. The first one, it is that is an epizootic pathogen. And an epizootic pathogen is something that has been introduced that is not from the country, and it has one point of origin, and then it just moves like a wave through the environment. And this hypothesis suggests that BD was actually introduced in the US in 1949 by the African clod frog, Sanovus levis, when they were using it for pet trade, and it was also being used in laboratory experiments. Now, the best example for an epizootic pathogen, we have it right now, it's coronavirus. So you can see that it spreads very rapidly, and you have high peaks of infection, and everybody gets infected because everybody's a naive host. In the case of BD, we see this mass mortalities, and one of the most iconic places to look for them is in Sierra Nevada and California, where the populations of yellow-legged frogs have declined very, very rapidly. The opposite of an epizootic pathogen is a enzootic pathogen, which means that the pathogen actually belongs or has been in the area for a while, and you have different points of outbreaks at different periods in time. And it could be due to changes in the environment or fragmentation, also changes in the community composition of the population. Or if you introduce a new species, something like that, that causes those outbreaks. So if you compare it with our current situation, it will be kind of like the flu, that it only happens seasonally, it doesn't affect everybody, and the curve for that pathogen has been flattened pretty much. So that is what an enzootic pathogen is. What we wanted to evaluate was, first of all, how long has BD been in the US, and whether if it was actually episodic and brought during that pet trade, or if it has been here for a longer time. And the way we did it is we actually went to the BRTC collection here at your university, and we collected 30% of species per decade. So I selected 8 different species that are actually widespread throughout Texas. They have very wide distribution. And from those species, I sampled 30% of the 30s, 30% in the 40s, 30% in the 50s, and so on and so on. because I mentioned before that this is a skin disease and you actually put the animals in a collection all in one big jar. What we did was we washed them thoroughly with ethanol, and then we took a skin swab. And that skin swab we took to the lab, did DNA extraction, and used a real-time PCR protocol to detect the presence of the pathogen. This is some of the frogs that we were using for that experiment. In this table, I'm actually showing you all of the species that we collected. The ones that are highlighted in yellow, Hylocyneria, which is a tree frog, and those three toads, Bufo speciosus, woodhousea, and debelis, didn't actually have any positive amplifications of the pathogens. We exclude them from the study. And the ones that I'm showing you in the green table, Lithobates catesbianus is the bullfrog, Acris crepitans is a cricket frog, Lithobates berlandieri is the Rio Grande leopard frog, and Hylover cycolor is a green tree frog. Those are the ones that actually presented positive amplifications. So overall, we had 859 samples. From those, 116 were positive. That gives us an overall prevalence of 13.5%. And the species that had the highest prevalence was the bullfrog with 17.9%. And the one with the lowest, as you can see, was the gray tree frog or the Hylover cycolor with 2.9%. This is the distribution of all of our samples that we collected. Anything that you see in red is a positive amplification for the pathogen. And mind, this is within the 30s all the way to the 2000s, right? Anything that you can see in negative, it's obviously everything that is black is a negative amplification. If you can see more, everything that is focused around central to East Texas, we have actually more samples than we do in West Texas, but we can still find some positive samples in West Texas. So even though there's a little bit of sampling bias just because of how all the samples have been collected throughout the years, we still had enough to collect positive amplifications from West Texas as well. And there seems to be nothing positive in the Panhandle, at least in my sampling. This table shows an overall prevalence of BD per decade. So again, we have one amplification in the 30s, but you can see how it's steadily, everything that is yellow, it's a positive amplification. And you can see how it actually steadily increases. And we have our highest percentage being in the 80s and 90s, which is pretty much that curve that you can see in white. And then it drops a little bit in the 2000s. After that, we wanted to do a Krigan analysis, and pretty much what this map is showing you is different points of outbreak and how this model will suggest the pathogen will spread. So if you look at that bright red dot, that represents the 1930s and 1940s, so the outbreaks that we saw then and how it starts moving in the 50s and 60s, how would the pathogen be dispersed? And as I mentioned before, even though we have one point of origin because of our sampling, you can see different points of outbreaks throughout the decades. You see more in Central Texas around the 60s and 70s. You can see another point of outbreaks in the 80s all the way down at South Texas. And overall, what the model is suggesting is by the 2000s, the entire state will have at least some sort of amplification for BD. But given this analysis, we actually did something that is called a spatial clustering analysis, and you can't see it because of this. But what we did was it creates clusters based on how many positive it would expect, and it deems it a hotspot if you have more positive than what the model is expecting to, which means this could be a good place for finding an outbreak of the pathogen. And we have two significant hotspots. One is cluster 1 in Central Texas, and one is cluster 2 in East Texas. This could possibly be due because of, in Central Texas, the distribution of all of the 4 species that we were studying overlaps. So anything that is a leopard, Rio Grande leopard frog comes from West Texas all the way to Central Texas. That's its distribution. Bullfrogs are all over Texas. cricket frogs actually are from Central Texas all the way to East Texas, the same as green tree frogs. So Central Texas is right the spot where all of the species actually get together. And well, if you think about it, the more swampy the region is, then the more likely you have to have more frogs, which means that everything that is from Central Texas to East Texas will probably have more frogs and potentially more possibilities for a contagion than if you're actually in West Texas. The opposite of a hotspot will be a cold spot, where you're supposed to have positives, but the analysis or the samples that I have didn't show as many positives. And one of our most prominent cold spots here would be Cluster 5, which is all the way North Texas, and then Cluster 6, which represents pretty much all the area where Dallas and Houston and all the major cities are. So this is probably why this is a cold spot, is because at this point there are not many frogs in such a urban environment. So what do we know? We know that BD was actually first detected in the 1930s. This is the first detection in Texas for this pathogen. In the 1940s, we have the first detections for these two species, cricket frogs and Rio Grande leopard frog. That's the first detections for the US. The same happens with Hyla versicolor in the 60s. We know that in the 80s, the prevalence reaches peak with over 30%. And right now, the current prevalence is 13.5. We know that our temporal distribution, the map suggests that BD might behave like an enzootic pathogen. Remember that we have different points of outbreaks at different periods of time instead of just one big movement. And our spatial analysis suggests that the most significant places to probably cause an outbreak of this pathogen will be Central Texas and East Texas. And finally, I would actually like to mention the importance of this type of collections that actually allow me to do all this work without a lot of traveling, and we can actually backtrack all of the species all the way to the 30s, which is actually pretty great. With that, I would like to thank Dr. Toby Hibbets for allowing me access to the collection, my friends that helped me swab all of those frogs, and both the universities that helped me with this project. And I'll take any questions.
Thank you, Andrea. Now you can make questions. You can use the chat to make questions or unmute yourself. And we have some minutes for questions. We have about 3 or 5 minutes for questions.
Hello, Andrea. This is Rose.
Hello.
I just wonder, because you see the trend have been increasing, and we can see right now the prevalence level also increasing like every 10 years. So do you have any hypothesis that that could be the case?
I think part of it, obviously, it's there's some degradation of the DNA when I collected from the samples. So there might be that I didn't find as much BD in the 30s as I would have found it, in the 2000s. I think that's part of it. I also think that part of the increase is that habitat fragmentation, right? In the 30s, animals had more space and they weren't so close together. And I know that social distancing is something that works for all of diseases, not just coronavirus. So if we force all of the animals to live in the same ponds because they don't have space, because we've been pushing it out, then this creates a higher prevalence where we find them. Environmental changes might cause this too. BD is a very aquatic pathogen, so if we're reducing the places where they can actually be, there's actually so many places where the pathogen can just live. So all of the animals are going to be in those places. There could be a little bit of sampling bias as well, just because of the places we're collecting animals. So if we had more animals probably from West Texas or the Panhandle, that will probably reduce the prevalence in some of the years, which I think it's that drop that we see in the 2000s. it might be that it's not a 30%, a 20% drop, but it might be just that, you know, people stop collecting. They have enough frogs as it is all the way up to the 80s. So that might also cause those biases in the sampling. I would think those probably are the causes for this.
Thank you, Andrea. And I want to let you know, my 6-year-old daughter, she watched this presentation with me. So I really appreciate the visualization of your presentation. So I also wonder, do you have any suggestions for like elementary school kids? How can they protect amphibian species?
Whoa. I think the challenges that amphibians in general being affected here in Texas more is actually try to keep the water and all those places not polluted, and hopefully we could keep some places that are for them, right, that we don't urbanize it. That would be great. So in general, if you're in elementary, just try to clean the waters, like get the schools to go out and, you know, pick up the trash from places, and create little ponds in your houses if you have the space for it. That would be great.
Thank you.
Does anyone have any other question?
Nope.
So thank you, Andrea. Now we're gonna have a little break until 2:50 so we can have our next talk. Thank you.
Yay. Thanks.
Now we're gonna have the second talk of this afternoon for plant and microbe session. We have now Caroline Wilson to talk about some volatile and plants in cotton plants. So welcome, and now it's your turn to talk, Carol.
All right, hi everybody. I'm Caroline, and I'm going to be presenting you some of my research that I've been doing for my undergraduate thesis. And so I've been working on volatile organic compound emissions from flower buds and fruits of cotton plants treated with beneficial fungi. All right, so fungoendophytes are microorganisms that live in plant tissue, and they've been shown to have many beneficial abiotic and biotic effects, such as drought and heat tolerance, and they have also been shown to limit pathogen in plants and herbivorous insects, so they can be helpful in agriculture in this way. And they've also been shown to affect insect behavior and performance, in that they can repel some insects, or sometimes they attract certain predators. And volatile organic compounds are a form of a plant's direct defense. So they are basically a plant's odor, and they can repel certain herbivorous insects because they are sometimes toxic or have toxic chemicals in them. And in previous studies, they've shown that there has been an increase in volatile organic compounds that have been treated with fungal endophytes. And this study was done in lima beans. And then in studies with red clover and tomatoes, there have also been shown to be increases in volatile organic compounds in the field versus when the plants are in a greenhouse. And this is some past research done with fungal endophytes on sucking bug responses to cotton reproductive structures. So these reproductive structures could be squares or bolls. So the squares are the flowering part and the bolls are the fruit. And in this study, they used two different fungal endophytes. They used Beauveria bassiana, which is a commercially available fungal endophyte that has shown to have entomopathogenic effects. And then the other one is Phyalomonium inflatum, and that endophyte came from a surface sterilized cotton leaves as part of a survey of naturally occurring foliar fungal endophytes. And basically in this study with Sword et al. 2017, they had these sucking bugs, and they were doing a choice test, and they found on the bottom, you can see there's the endophyte treatment, and on the y-axis is the latency of first contact, so how long it took for the insects to contact the tissues. And you can see in both species of sucking bug, the treated tissues tended to have a longer time before they were contacted by the bugs. So with this in mind, I wanted to run some tests, and these were to test whether cotton plants treated with these fungal spores would influence the VOC emissions from reproductive structures. And then I also wanted to test whether greenhouse or field growing conditions would influence the VOC emissions from reproductive structures. And I did this because in that Sword et al. 2017 study, they hypothesized that the volatile organic compound emissions could be what was the thing that made the bugs not contact the plant. And they also in that study used both greenhouse and field structures. So with my experiment, I started with sterilizing some seeds, and then I treated them with the Beauveria bassiana and Phyalomonium inflatum spores at a 10 to the 7th spores per milliliter. And I also had a control, so there were 3 total trials, so to say, if it's control, Beauveria, and the Phyalomonium. And so then I soaked these seeds overnight before planting them. And basically with the volatile collection, I had 3 trials going on. I had a greenhouse square trial, a greenhouse bowl trial, a field square trial, and a field bowl trial. And within each of these trials, there were 30 samples, 10 of Beauveria bassiana, 10 Phyalomonium inflatum, and 10 control. So in this kind of flow chart that I made, you can see all these trials with their samples. So once the reproductive structures, either the square or bowls, were either, for the squares, I picked match head size squares, and for the bowls, I picked dime size, or quarter size bowls. And then I would excise for the squares 4 squares, or for the bowls 3 bowls, because they have more, bowls are a little heavier, they have more biomass, so I used a smaller amount. And then I'd incubate them in a jar for 1 hour, and then I would do a 30-minute solid phase microextraction sample, and then I would insert the fibers from the solid phase microextraction, the SPEME samples, into the gas chromatography for analysis of the volatiles. So when I did this analysis, I found all of these compounds, a mixture of terpenes and green leaf volatiles, which are all defensive compounds that you can commonly find in plants. And you can see there's a lot more terpenes than green leaf volatiles. So when I used this, when I looked at the gas chromatography, I got some chromatograms that I used to look at the, not only this is the number of volatiles that I have, but the actual amount. I could get a quantitative result from this. So looking at all these results, I did a permanova in R with some stats using the function Adonis and the vegan package. And what this permanova looked like is in this chart here. You can see that for looking at, when you're looking at the volatiles and you look at the location and structure and the interaction between location and structure, this was significant. There was a significant difference basically in the field or the greenhouse, and also whether we were using bowls or squares in the volatile organic compound emissions. And you can see that treatment to the fungal endophytes did not have a significant effect. And continuing looking at these results graphically, you can see that each of these boxes represents one of the trials, and all of the shapes connected to the box are the individual samples. And what was interesting about this is that the greenhouse bowls and field squares seem to cluster a lot. And I think this could be because in the field, the plants are exposed to a lot more biotic and abiotic stressors. So they have to, a square is the younger reproductive tissue, so they have to get their defenses ready earlier than in the greenhouse. With an older tissue like a bowl, doesn't really have any sort of stressors, so it doesn't need as much defense. And so overall, their location and structure had a significant impact on volatile composition, and the treatment did not have a significant impact. And I'll go into maybe why that is. So one of the reasons I think that the treatment might not have had a significant impact in this particular study is that the sample size was relatively small. I did a lot of trials. I spread it out over those 4 trials, but within them, there were only 10 samples of treated product per treatment. So maybe if I had made a larger sample size and really focused on one grouping, then the treatment, I could have found some differences. And then the other issue could have been that I didn't weigh the exact biomass of the bowls or squares. I just used the match head or quarter size diameter. So maybe next time I would weigh that so I could be a little more accurate with those measurements. And in the Sword et al. Study, where they said that volatiles could be what is influencing the insect behavior, my study didn't find that to be the case, but like I said, maybe with a larger study, there could be significant findings, and it's possible that there are other effects besides volatiles that are impacting the insect's behavior, and that's something that we can continue to study in the future and see what all the effects are of the fungal endophytes. And with that, I'd like to thank everyone who helped me out with this project, and I'm happy to take any questions.
Thank you, Carol. So now everybody's allowed to unmute yourself if you have any questions, or use the chat to make questions. So Carol, what are the implications? How can you use the volatiles as a IPM strategy? Do you know something about that?
Well, I think you can use them to, if you know what you're doing with them, and you can, if you know, like with the fungal endophyte treatment, that you're having a certain increase in specific terpenes or green leaf volatiles, and you know that these they have been studies that do show that they affect insect behavior. So if you know exactly what you're altering, then I think maybe you could do treatments that make one that repels more prominent in your plant. So maybe that could help with reducing the pests you have. Or maybe even if you had some volatiles that are attractive to certain insects that are predators, and you could kind of use indirect and direct defenses, I think that has potential, but I'm not really sure how you could do that right now. But I think it's something definitely to look at continuously.
Okay, thank you, Carol. Luke made a question pretty similar of my question. So he said, thank you for presenting, Carol. In your opinion, what are some practical industry applications we can derive from your study?
Yeah, I think that's kind of the same thing as just trying to understand what fungal endophytes do, and especially since like Bovero bassiana is a commercially available treatment that you can use, understanding how it affects plant volatiles, and I'm sure it affects other things too, but once you really start understanding the exact effects, you can really hone in on how you want to use it defensively against pests, I think.
Okay, does anyone have any other question? Okay, thank you, Carol.
Thank you.
So now we're gonna have a break until the next talk that will be at 3:10.
Do you see me? It's okay?
Yeah, it is. You just need to really open your presentation so we see just the slides.
Okay, yours.
Yeah, it's working.
Okay, then you say me if I can begin.
Okay, I will wait until 3:10 so I present you and you start talking.
Yeah, thank you.
So now we're gonna have Juan Valdez talking more about prickly pear cactus. And remember that you just ask questions on chat during the questions time, so you don't bother the speaker. And you can both unmute yourself or make questions on the chat. So thank you, Juan. Now it's your turn.
You're welcome. Good afternoon, everyone. How are you? First of all, I want to thank the organizers of this great event of VEIS for their invitation. To all of you, thank you very much, and I hope you're okay and your families. So, my name is Juan Arredondo Valdez, and my advisor is PhD Alejandro Luna Maldonado. I present physical characterization of mature parts of prickly pear cactus. We're from Monterey City in the agronomic faculty. And I speak about the prickly pear in Mexico. The production in Mexico is 825,000 tons per year. And we have 20,853 hectares cultivated. And we can see in the map where are the most important producers of the crop. Mexico City, State of Mexico, State of Hidalgo, and Morelos. This picture, we can watch a Florentine codex where we can see the people using the prickly pear in ancient times, maybe 1540, 1585. This is about the general history of things in New Spain from Fray Bernardino de Sayagon. The uses actually of the prickly pear in Mexican culture is food, medicine, and nutrition. Like a medicine, the lower blood sugar levels due to the high content of fiber or mucilage that the nopal has. So prevents the development of osteoporosis, controls gastritis, and provides nutrients such as calcium, protein, carbohydrates, iron, and ascorbic acid. Thanks to the great capacity to adapt to the difficult conditions of the semi-arid and arid areas of the world, and the forage, nutritional, and healthy use, the nopal is considered an important crop in the investigation of relevant information to make its use more efficient, thus like conserving the current gene pool. In morphology, the morphology and colorimetry are important agronomic characteristics in product seeds. Science, they affect yields. In obtaining this data accurately, many measurements are necessary due to the irregular shape and similar sizes of the parts of the plant. There are a lot of background about some studies in morphological characterization. And the acquisition, the processing and analysis of the images of the plants have acquired much importance for selection and taxonomic studies because have close association with freshness, with maturity, with variety, convenience, and food safety. Our general objective of this part of the project is to research the discriminatory potential of Oponia ficus indica mature parts, measured accurately using image analysis and colorimeter. Our materials, we use cladots of nopal, 1 year old, of the Villanueva variety, and were selected and collected by people with experience in the management of seed slides of the Alejandra farm. This is the site. And then they were taken to the agronomy faculty. This is the place of the experiment. We can watch the place. We put 30 plus 15 more cladots in 22-liter pots. Then we collect the data. We measure the color parameters in both sides. Then a cloud photograph with a reference scale, that's important. And measure weight and temperature. For the morphological analysis, photographs of each of the three cladodes were taken and processed with the Mesh G software, considering 12 parameters. Here is the flow diagram of the process. We can watch the diagram, the flow chart of the image acquisition process. We open our file, but this is very important to set up scale from the centimeters or millimeters or nanometers, etc., to pixels. And this is the final form. In this case, our cladode is the product, but you can use another product. Using image G software. We can watch the main menu of the software. By our hand, we obtain the colors of both sides of the cladodes, considering the mean value of the sides and average of parameter reading. We take 4 or 5 months each week, the color, the parameters. We obtain L, B, C, L, H parameters. Then in this card, I show the mean values of the color attributes of versus each one of the cladodes. We can watch the more or less is similar, but has some variation about the three cladodes. The luminosity ideal, the values, the hue, and other parameters. Here we can watch the table 1, the mean values of L, C, and H color parameters. And using a program of Johnston 2019, we can watch our values like a color. I put the L luminosity 47, the C 17 more or less, and H 122, and look the color. Then the statistical analysis, a total of 5 quantitative colorimetric variables were analyzed at ABCL in both sides. Then we made descriptive statistics for the 12 morphological variables in 3 cladodes. So the analysis of variance, correlation, and nonlinear regression analysis was made. In this table, we can watch the mean values, the minimum values, the maximum values, and the standard deviation of all mature paths. We can watch the parameters. We analyze areas, XM, GM for the centroidal, perimeter, width, height, circularity, ferrite diameter, kurtosis, area relation, roundness, and solidity. And then we analyze it and obtain some correlation analysis between that morphological parameters. And obtain models, equation models of with a nonlinear relation using the curve expert software. We work with 95% of confidence, and we show the best values only for each one of the model. Okay, this is the same for each one of the pair of parameters. In this table, we can watch the related variables in each one of them. And the proposed model, like sinusoidal, Dr. Hill, Richardson, with the best values of the R values, with the best only. Our results at this time, we can say that with a process summary, but we reduce the time and labor involved in capturing and analyzing images of mature parts of Opunia to obtain quantitative variables related to external morphology of them. These variables were obtained by image processing using ImageG software and color instruments. Later, were fitted with linear and nonlinear models using Curve Expert software. And like a data summary, we can we have the values. Mature parts weight were 682.95 to 1,299 very variables, and then other values that we watch in the table too. The coefficient of variation varied from 0.75 to 7.58%. However, most of the variables showed a coefficient of variation of less than 10%. Like conclusion, concluding, very good correlation coefficient values of the nonlinear regression models were obtained between each pair of parameters. Only the best were the each pair of parameters. 5 more with a middle value of the 21 parameters, all with a confidence level of 95%. The obtained results of the research show that the image processing and linear and nonlinear modeling allow to characterize the morphology and color of Opunia ficus sinica, el mature parts Villanueva variety gladots of a girol using ImageG and Curve Expert software. We hope that this research will be helpful protocol for those who wish to carry out similar color and morphology characterization work with mature parts of different varieties, or other seeds, or other products. Then that's all. Thank you very much. And I am open for the questions. Thank you.
Thank you, Juan. Now, who has questions can unmute yourself or make the questions on the chat. Okay, Juan, I have one question, but it's not related what you are showing about water stress. But can we use ImageJ for prickly cactus to see like some diseases if there is something happening? Because at some way I know that some diseases can have water stress on the plant. So I don't know if this will interfere with your analysis.
Yeah, the ImageG analysis has many uses. You can find many works about the use of the microscope, and then how I say the scale is very important. Then when you use a microscope to watch something, you have a scale. Then that's very good for you because it's more easy for work. In this case, we put a scale, external scale for relation. But yes, you can use very more in very much cases the software. I say that we are working with hyperspectral analysis now, but this is as a part of only of the work. Okay, but the ImageG is good, I think.
Okay, thank you. Does anyone have any other question? Okay, Juan, thank you so much. Now we're gonna have a break until our next talk. That will be at 3:30.
Thank you very much.
Thank you so much.
Bye.
Okay, now we're gonna have the last talk of the day of our session from plants and microbes, and we're gonna have Ashish Nerlekar. Sorry if I mispronounced your name. Talking a little bit more about savannas. So now it's your turn, Ashish. Thank you.
Hi, just a sec. Yeah. Okay. So, hi, I'm Ashish, and I'm going to present a talk which will tell you a story about the ecological history of a tropical savanna in India. Okay. So, the dominant narrative of the past century or so has about tropical grassy biomes, which includes grasslands, savannas, and open canopy woodlands, has been that they are pretty recent in origin. And that means that they're pretty much formed as a result of human-induced deforestation. So because of this, they have been widely misinterpreted as arrested successional stages of a forested ecosystem, and more so for the Indian savannas, which have been long assumed to be a product of human-induced deforestation. So this is the key background or the key problem on which I'll be basing my study. So with that, my question for today's talk, which I'll be addressing, is, are the savannas in Pune city, India, anthropogenic in origin or not? So these are my hypotheses. So if they are actually anthropogenic in origin, we would expect something like this on the top panel. So we would expect that savannas would have formed after a forest configuration, which means that at some historical state or point in time, there would be a forested configuration with closed canopy tree species without a grassy continuous understory layer, being replaced by a savanna configuration with scattered trees and a continuous grassy layer in the understory because of anthropogenic degradation. But if these savannas are actually not anthropogenic in origin, which means if they're ancient, we would expect to find this savanna configuration no matter how far back in time we go. So the same throughout. So these are the two hypotheses we'll test. So I'm sure that at least some of you were surprised to hear that the savannas in India. So here's a map of where they are. So the shades in blue are savannas in Africa and Asia, and this triangular portion in India over here is the savannas in India, and this is Pune city, which will be the study location. So in order to test these hypotheses, I'll be using data sources from these four themes, and I'll be summarizing them to find out, so to try and test these hypotheses. So this is how a part of the hills looks today, with scattered acacia trees and a grassy continuous layer. This is time zero, so as of now. What we'll do is in the next slides, we'll try and go back in time and see if we find a forest ecosystem sometime back. So I present my first line of evidence, which is historical records. And this is a picture of the same hill which you saw in the earlier slide, taken about 100 years back from today. And you see that there's clearly no forest 100 years back. So it's the same savanna configuration. Let's go back a century further back. And this is a painting done somewhere around 200 years before present. And it shows that in the bottomlands where you have civilization, you have these trees which are clearly planted, but the uplands, which are the hills, these clearly represent open ecosystems with shrubby and grassy layer. So clearly no forest. We can go back still a bit further to find that this painting again shows a clear savanna configuration, so no forest. The limitation for historical records is they do not take us back beyond a couple of centuries. So we need another line of evidence to look at that, which is paleoecological records. So this is the second line of evidence, and this is essentially paleo or fossil pollen records from a place in central India, which is biogeographically similar to the Pune city savanna landscape. So in the left panel over here, you can see the y-axis represents years before present. So 0 years, which is today, 1000 is 1000 years before present, 8000 years before present, so on. So from top to bottom, you go back in time. This is a panel which represents the abundance of pollen grain for these three functional types of groups, functional types of species. So the first is moist species, which are essentially forest species. The second is the savanna species, and third is the scrub species. So the agricultural expansion in this region in the western part of India started only since 4000 years before present, which would have been a major source for forest or land clearance. So 4000 years is this line over here. So if our narrative was true, we would expect a savanna signature from 4000 years to today, which is this part of the graph. Sorry. So we would expect a savanna signature from 4000 years to today, which is the top part of the graph, but not so in the bottom part. So we would not expect to find savannas right in this region. But you can see clearly that savannas are present throughout this entire spectrum, and they actually spike at around 9000 years before today. This panel on the right, you can ignore whatever's written in there and just focus on the colors. So the reds indicate drier times in the subcontinent's history, whereas the blues represent wetter times in the subcontinent's history. So you can see that wet zones correspond well with the abundance of moist species. Dry times correspond well with the abundance of scrub species. But irrespective of all of this, the savanna element dominates up till 9000 years. And this is where our pollen records can take us. But my next line of evidence takes us back millions of years. So this is a phylogeny of a group of lizards called the fan-throated lizards, which are found on these hills. And the x-axis over here is million years before today. So this is 0 million years, 10 million years, so on. And you go back in time over here. And this graph represents the diversification pattern of this group. You can see that so before that, the green part indicates wet regions in the Indian subcontinent's history, and the transition to ochre zones means drier parts or aridification. So it's very clear that this Sitana group has diversified somewhere around 5 to 10 million years, consistent with the aridification of the Indian subcontinent. So the presence of such species is another indication of an open landscape. So these are two more examples of another group which shows a very similar pattern, presence of species which diversified in response to the late Miocene aridification. And the last line of evidence which I'll present is the characteristics of the present or extant biodiversity. So as of now, there are quite a few endemic plants like this dwarf Jatropha with tuberous underground parts, which are a clear adaptation for frequent fires. C4 grasses like this endemic Lophopogon, which have evolved way back around 4 million years ago, and a range of other endemic diversity, especially reptiles. And people are still documenting more endemics from this region. So if this was a forested region before and then a savanna, we would expect to find forest generalist species instead of savanna endemics. So that's it. And with this, I would conclude about my hypothesis that this region is indeed antique, which means that these savannas in Pune city are actually really, really ancient. So you can see in this lower panel that they were clearly as they are today with the savanna configuration since at least 4 million years, and have been maintained by regular surface fires and grazing throughout time. So imagining such ecological histories is pretty common around the world. So people have imagined such forested histories in many parts, including areas in Africa where people actually thought that open spaces created around some villages were anthropogenic, and then these notions were also dismissed recently. So what does these imagined histories mean for our land management today? So in this image, you can see trenches dug for planting trees in this region with the aim of reforesting the region. But reforestation would mean that there was a forest before, and now we know that there clearly wasn't a forest. So this clearly can be termed as afforestation. And research is pretty clear that planting trees in regions where they do not belong does more harm than good to the biodiversity and ecosystems, which ecosystem services that these places provide. So with that, I'd like to leave you guys with a single important message, which is savannas like forests can also be ancient, and that appreciating their antiquity is critical in the light of global change. I'd like to thank some libraries, my funding agencies, and friends and experts for this, and I'm happy to take questions.
Thank you, Ashish. Now we're gonna have time for questions. As I told you before, you can unmute yourself or make the questions on the chat.
Hey Ashish, I just have a doubt. So do you think the same trend would be there in all savannas across the world? Or like, do you think the difference in the trend would be between the city, the urban savannas and the savannas which are like a bit less disturbed?
Right, so that's a good question. So the thing is, a lot of socio-ecological things also matter because these are savannas within the cities. But if you talk of the landscape in general, this clearly applies to a lot of other tropical savannas, especially places like the Cerrado of South America, which is a mega diverse region, but people thought that it's just a degraded forest. So it does clearly apply to a lot of other places, especially in the tropics.
Oh, sorry, I guess I have some network problem.
Okay, should I repeat the thing or?
Yeah.
Should I repeat it?
Yeah, maybe that last one.
Okay, yeah. So I guess it's pretty common that people have misinterpreted tropical savannas to be degraded forests, is what I was saying. So it's not just India, it's not just some countries, it's clearly most of the tropics actually. So it does apply to a lot of other places.
Okay, thank you.
Does anyone have any other question? Oh, there is one on the chat. That was a fantastic presentation. Ashish, what are your recommendations as far as land management goes? Not just in the ecological sense, but in a broader societal setting.
Right. That's a really nice question. So ecologically, it's pretty meaningless to add trees. But in the sense of society, in the sense that these savannas are located in a city, it's pretty tough to handle this thing because we have been brought up with this notion that trees are good, planting trees is beneficial for so many things. And what needs to be done is actually trying to demystify this thing that trees everywhere are good. So it starts with education early on, is what I think. And uh, appreciating savannas in an urban setting is critical because these are the places where you have most of your decision makers. So if you convince decision makers in your city that, hey, see, those hills, they're really ancient, you can probably convince them to save other places which are not so accessible to people as well. So that's my take on it.
If you have any follow-up questions, look, you can unmute yourself so you can keep talking to Ashish. Does anyone have any other question?
I have one more question.
Yeah, sure.
Yeah. So do you think many of those species which lived like maybe which were there in the region some several years ago would be suitable to grow now with the current climatic conditions, because the climate has also changed with the due course of time over the millions of years?
Well, what we see is the species which have gone locally extinct are the ones which were savanna endemics or savanna open canopy species in terms of plants typically. So I wouldn't say the climatic fluctuations have been that severe that it would hamper restoring them back. Again, it's a huge challenge to restore savannas back. So it's an entirely another issue that if or how should we go about restoring things which are converted into plantations right now. So that's an entire ballgame.
Okay. But given the scenario, if I want to preserve the existing extant species which are still there, so what would you go forward? So do you think like you just keep it the same way, protect?
No, actually, the thing is, right now, throughout other places in the tropics as well, fires are seen as detrimental for the biodiversity. So it's a huge task to make people aware that not all fires are bad and that fires have been important at least since 4 million years in shaping these landscapes and the biota. So the moment you stop fires, which is happening everywhere in the world right now, you automatically lose out on those endemic species. So there's a lot of things we can do proactively. Also, because this is an urban setting, you can have a lot of um, flagship species for the regions and trying to tell people stories using those species and how their habitats were before, what they need, so on.
Okay, thank you.
Any other question? Okay, so now we can be done with our session for plants and microbes. Thank you everybody for coming and for participate, even if it is for a Zoom meeting. And everybody had a good attendance, and I appreciate that. Thank you so much. And we're gonna have our closing remarks with the awards at 4:30. So if you want to participate and see how it's gonna be, who it's gonna win for each session, it's important for everybody to be there. So thank you so much, and see you next year.